Gregor Mendel was an Austrian monk who discovered the basic principles of heredity through experiments in his garden. Mendel's observations became the foundation of modern genetics and the study of heredity, and he is widely considered a pioneer in the field of genetics. We are going to see videos that represent Mendel experiments on the field of genetics. He developed the model of heredity that now bears his name by experiments on various characteristics of pea plants: height, tall versus short, seed color, yellow versus green, seed coat, smooth versus wrinkled, etc.
Started out with plants that bred true, that is, when tall plants were self-pollinated, plants in following generations were all tall. When the short plants were self-pollinated, the plants in following generations were short, all short. We found that if true breeding tall plants are crossed with true breeding short plants, all the next generation of plants called F1 are all tall. He showed that self-pollinated F1 plants produce an F2 generation with 3/4 of the plants tall and 1/4 short. He discovered the laws of inheritance, the law of dominance that states that some alleles are dominant and others are recessive, and the law of segregation, and segregation is the separation of alleles during gamete formation. Mendel thought after his discovery that there was something altering the traits of each plant that he was working with. He didn't know yet that it was about genes. This influenced in society because it was explanation of why are we so different, and not only in our skin color, but also in our traits in the face or all in the body. Punnett squares are tools that help to show all possible allelic combinations of gametes in a cross of parents with known genotypes in order to predict the probability of their offspring possessing certain sets of alleles. Makes use of a grid and letters, capital letters to represent dominant alleles and lowercase letters to recessive alleles. With this tool, the known genotypes of each parent are shown to help predict the possible genotypes of their offspring. It shows how alleles are inherited or passed onto offspring from parents. It is typically applied in monohybrid crosses and dihybrid crosses in which the theoretical outcomes are based on the assumptions of segregation and independent assortment of alleles.